Take a hard look at these unique fish. They may not be what you're used to seeing at the end of a fishing line, packed in the frozen fish section of the supermarket, or shown in mainstream TV shows or movies with scenes about fishing. In fact, these fish are still considered trash fish by many confused anglers who unsuspectingly catch one, and they don't receive much attention outside the circles of conservation biologists or graduate students who are tasked with studying these species. Yet these ancient fish are special and, might I even say, beautiful. Flannelmouth suckers, bluehead suckers, and roundtail chub, which in the fisheries community are collectively known as the three species, are endemic to the Colorado River Basin and exist nowhere else on Earth outside the 7 states that encompass the basin. 3 species have evolved to withstand extreme seasonal flow fluctuations, intense periods of droughts and floods, high salinity levels and sediment loads, and highly variable water temperatures that characterize the waters within the Colorado River Basin. Once common in the main stem rivers and tributary streams within the Colorado River Basin, 3 species now occupy only about 50% of their historic range, and abundances are not as robust as they historically were. These sharp declines in distribution and abundance are mainly due to the onset of water development projects and associated dams and reservoirs, as well as the invasion of non-native fish species that not only compete and prey upon native fish, but also hybridize with native fish, threatening the existence of these species even further. Within the upper Colorado River Basin, in the southwest corner of Colorado, sits the Dolores River Basin. The Dolores River Basin is comprised of the Dolores and San Miguel rivers and smaller tributary creeks and streams that feed into these rivers. The only major dam within the basin is McPhee Dam that impounds water within McPhee Reservoir. There are roughly 200 free-flowing river miles below the dam before the Dolores River meets the Colorado River near the town of Cisco, Utah. The Dolores River Basin is unique when it comes to the 3 species fishery. It is one of the only intact populations of 3 species that isn't already heavily bisected by fish barriers and influenced by non-native fishes. The fact there is only one major dam that is complete barrier to fish movement within the basin means there's excellent connectivity among the Dolores, San Miguel, and Colorado rivers, giving 3 species unimpeded access to a diversity of habitats. In addition, the composition of native species when compared to non-native species is consistently high both spatially and temporally. The excellent connectivity paired with the relatively low abundances of non-native fish makes the Dolores River Basin one of the last remaining strongholds for 3 species in the world. Although it is known that the composition of native species when compared to non-native species is high, the overall status of 3 species within the basin is difficult to assess. This is because the composition and abundance of 3 species is highly variable both spatially and temporally, and large proportions of 3 species are highly transient, as evidenced by sampling in 2019. The San Miguel River was sampled to assess the fishery 3 different times in 2019, once in May, once in July, and once in September. 3 species composition and abundance varied drastically among the surveys, and in the survey in the fall, no 3 species were captured at all, while hundreds of 3 species were captured in the same reach during spring sampling surveys. Furthermore, quantitative comparisons of data are difficult because standardized sampling is extremely challenging to achieve in many reaches of the river because of lack of flow and accessibility, which means that most surveys are conducted under slightly different sampling conditions and flows, making it hard to directly compare data among years. Assessing an assembly of fish that are highly transient and challenging to sample consistently resulted in a knowledge gap of when, where, and how 3 species are distributed throughout the Dolores River Basin, both spatially and temporally, as well as which tributaries are being used by 3 species. One tool that can be used to assess movement and identify presence of animals throughout a landscape is passive integrated technology. Passive integrated technology essentially has 2 components, a transponder and a receiver, or an element that carries information and an element that receives information. For our research, the element that carries information is called a passive integrated transponder tag, or PIT tag for short. Each PIT tag has a unique code that is programmed into a microchip that is encapsulated into a small glass tube that can be implanted into organisms. When the tagged animal passes within close proximity to a special antenna called a passive interrogation antenna, the emitted frequency of the antenna excites the tag and causes a responding signal from the tag to be sent to the antenna to be read. Once read, an onboard data logger on the antenna keeps a record of the date and time each tag ID was encountered. A collection of one or more antennae is called an array. Arrays are especially useful when studying riverine species because they are able to cover the whole stream channel and allow for the determination of movement direction. For our research, we utilized 2 permanent stream-wide arrays and 8 smaller portable submersible readers. The first array, known as the Rio Mesa Array, is located 11.5 miles upstream from the confluence of the Colorado River and the Dolores River. The second array, the Disappointment Creek Array, is located 131 miles upstream from the confluence, in the upper reaches of the Dolores. These arrays were installed in the Dolores River Basin in 2013, and we were able to analyze stored detection data from their arrays dating back to 2015. The submersible readers were deployed in 8 different sizable tributaries throughout the basin from March until June in the 2020 field season. Utilizing the permanent stream ride array and portable submersible readers, coupled with pit tagging efforts that span multiple agencies and states throughout 8 years, we are able to investigate the movement patterns, large-scale use, and tributary use of 3 species within the Dolores River Basin. For the large-scale use and movement pattern investigation, we investigated the detections of pit-tagged fish on the stream-wide arrays in several ways. At each array, we analyzed the total number of unique detections for each species within each year, as well as the direction of movement associated with each detection. We also studied dispersal analysis, meaning we explored where fish were coming from that were being detected based on the original tagging locations of the fish. For the tributary use chapter of the research, we determined which tributaries were being used and by what species, and where those tagged fish were coming from that were using the tributaries. Our analyses revealed interesting and informative results regarding the movement patterns of 3 species within the basin at each different array location. At the Rio Mesa array, we found that large numbers of native suckers were moving upstream across the array in late March and early April each year. At the Disappointment array, the peak season of movement was later in the year, from May through September, and fish frequently moved both upstream and downstream across the array throughout the year. However, downstream movements were slightly more common. We were also interested in species-specific differences in movement timing and found that native suckers are accounting for the peak of detections on the Rio Mesa array, while in the Disappointment array, native sucker movement generally occurs from late April through May, while the movement of Roundtail chub are more concentrated towards the late summer and fall, from July through October. When analyzing detections of fish at each array based on their original capture locations, we found that large percentages of native suckers that were originally tagged in the San Miguel River and Colorado River were detected on the Rio Mesa array. With all tagging efforts combined, roughly the same number of fish were tagged in the Colorado River Reach and the San Miguel River Reach, and the percentage of those tagged fish that were encountered at Rio Mesa were nearly identical. Yet the fish tagged in the Colorado River Reach were tagged at the most 25 miles away from Rio Mesa, while the fish tagged in the San Miguel River Reach were tagged at the least 55 miles away from Rio Mesa. This highlighted the importance of maintaining connectivity between the Colorado, Dolores, and San Miguel rivers, and also indicated that 3 species tagged in the San Miguel River are highly mobile and move long distances. In the upper reaches of the Dolores River, on the Disappointment array, the majority of 3 species being detected were originally tagged fairly close to the array. However, in 2019, a year characterized by high flows in the Dolores, over 40 Flannelmouth suckers were detected coming all the way from the Colorado River, a distance of over 130 miles. As for the tributary usage research, we found that 4 of the 8 tributaries with submersible antennae detected fish, and the fish being detected move long distances from their capture locations. Flannelmouth suckers and Bluehead suckers originally tagged in the Colorado River were later detected on West Creek, Mesa Creek, and Tabawatch Creek, which is 70 miles from their original capture location, while several roundtails captured in the upper reaches of the Dolores were encountered at La Salle Creek, a journey of about 30 miles. This research has important management implications, and the information is already being used to inform land management decisions to improve the fishery. For example, the movement pattern and large-scale use analysis is being used to inform the design of a soon-to-be rebuilt diversion structure that will incorporate a fish passage structure. By identifying tributaries that are being accessed by 3 species, fisheries managers are now working with landowners to improve connectivity to important tributaries, such as the redesign of an old diversion structure on Mesa Creek to allow for fish passage. Overall, the increased understanding of when, where, and how 3 species are utilizing the Dolores River Basin as a whole has set the stage for improving and informing management actions in an effort to conserve one of the last 3 species strongholds in the world. I would like to thank the Southwest District of the Bureau of Land Management Fisheries for funding and supporting this project. Thank you to Colorado Parks and Wildlife for the time and resources they have provided throughout the entirety of this project. I'm especially appreciative of my faculty advisors and committee members at Western Colorado University and beyond who have provided much needed assistance and guidance with all aspects of this research.